Hello students, welcome to the first part of lecture 2 of week 11. So in this part we will discuss what is all about like gene prediction in prokaryotic genomes. Eukaryotic we have already done in the earlier classes, right? We will see the coding genes, protein coding genes, as well as the RNA coding genes, such as which are typical for a new genome, such as t-RNA genes and ribosomal RNA genes, right? And also like methods which can be used for the function annotation of any bacterial genomes, and or basically or a set of genes derived from bacteria, right? So basically what is a gene? It is a piece of DNA that is coding, right, for a function. The function could be structural function, regulatory function, protein coding function, right, in the form of proteins, right? And basically what is there in the gene will be expressed, and that will be responsible for the phenotype also that you observe, or the properties or characteristics of the genome, right? So gene is something that is required for the functional profile of a organism, and it has a structure. That means if we like I assume that you all have done your molecular biology courses and genetics, okay? So you know the structure of gene or have some idea about it. So based on that biological or theoretical knowledge that is we have got in the books, we can translate that into programs which can be used to predict genes and also later for the annotation. Okay, so basically for gene prediction happens after you have got a new genome, or there is a gene that was unannotated and you want to find the function of that gene, right? Or you have a set of sequences for which you want to classify them in terms of function, pathways, or ontology or orthology based annotations, right? And then later put them into pathways and like. So basically the commonly used gene prediction program for the prokaryotes include Glimmer, which is the most common, Prodigal, GeneMark, which was earlier very properly used, but still you can use, and that has some merits, right? And a very common approach which is called ORF finder, that we will also discuss in this lecture. So basically if you see the DNA, it can be read in 6 different reading frames, and okay, because it is double stranded, so in the first reading frame you can if you shift the codon by 1 base pair, which is 3 base pair codon, okay, you can end up having 3 different codons from like 4 or 5 nucleotides, right? So like in this case, if I start from here, I can read this as AGG or GGT or GTG or TGA or GAC, right? And all these will code for very different amino acids, and same for the reverse strand. So potentially you can have a reading in 6 different reading frames, and suddenly you may see that okay, at this position I got a stop codon, and your basically the reading frame or a stretch of nucleotides arranged in codons will suddenly stop, and that could be a possible stop codon or could be an abrupt end for that small reading frame. And the relevant ones or long enough are called ORFs, and others are generally not taken into consideration for protein coding genes particularly, right? So these are like 6 different reading frames that you can got from this AGG, TGA, then shift by 1 base pair, okay, GTG and like that, okay, GGT, and okay. So this is what we explained. So and basically you can see that in the first reading frame it was coding for M, okay, N, E. In the second one, when I shift by 1 base pair, it coded for W, T and K. So it is very different in the 6 reading frames, and that is why a gene will always be in a single reading frame among the 6 reading frames. Okay, another representation of a 6 frame translation. Okay, so you can see that this is a double stranded DNA, and on both sides or from the forward or reverse strand you can get the 6 different reading frames. Okay, so now what is an open reading frame? So among the 6 different reading frames, there could be a stretch which is starting from a start codon, ending with a stop codon, and in between there are in the same reading frame, okay, that means 3, 3, 3, 3, 3, we are increasing or adding or proceeding further by adding 333 codons, or reading the DNA without leaving any gap, okay, because if you keep a gap or skip a nucleotide, it will be a different reading frame, okay. So continuous stretch of codons, okay, in between a start and stop codon, okay, will be an open reading frame, and the length of this will matter. So there has to be a minimum length that you would consider, okay. Similarly, like presence of promoter and other sequences in front, okay, or ribosomal binding site, that could be additional signatures for this open reading frame to confirm that this could be a gene, okay. And basically the gene confirmation happens when you get a function for this sequence, right. So gene prediction can be done by evidence based or ab initio method. Evidence means that we have already sequenced so many genomes, have data for genomes, genes, proteins, right. So we can take a new sequence and align against those genomes, okay, and see that by that comparison, if you find that there is a sufficient similarity and identity with that particular gene, okay, in the database, so that means whatever that gene in the database is doing the function, your gene may also be assigned to that function, okay. So that is like evidence based, that doing basically more of similarity, right. And ab initio is that by looking at the structure of gene, that okay, like gene has these kind of structures. In eukaryotes it has exon, intron, canonical splice sites and UTRs. In prokaryotes it is a continuous stretch ORF between start and stop codon, promoter, RBS, and these kind of signatures. So basically those can be used for the ab initio prediction of gene, which are basically done by program, and they also give a score to that gene. Okay, so now so far, like the previous discussion that we had was mostly about the protein coding genes. So there could be the RNA genes, other like t-RNA, ribosomal RNA, structure RNA, small nuclear RNA, or basically microRNA, right. So long non-coding RNA, and many different kinds of RNAs which are basically now gaining a lot of importance, and the protein coding, which is the messenger RNA or mRNA, okay. Prokaryotic genes are generally easier to find or predict because of their not so complex structure, whereas the eukaryotic genes finding may be challenging because the exons are split, and there could be a very long or unpredictable length of stretch between the two exons, or alternate splicing, and those things also complicate. Okay, so various approaches like direct approach could be one is finding the direct evidence of gene, like you have done the mRNA sequencing or found the ESTs or complete RNAs, and those can again be mapped on the genome, and basically that could be the gene, okay. Or comparing with a closely related genome, or finding a identical or like highly similar gene in the database, okay. Those are indirect. Is basically you are looking for something that looks like a gene, or you are predicting a gene from scratch, right, ab initio that we discussed earlier also, right. And sometimes like there could be two highly similar genomes, like two strains of E. coli, okay. And so like if the genome sizes are also like similar, and there are two different strains of E. coli, so you can say that most of the places where the known strain of E. coli has genes, at similar locations you may also find the other strain to have genes, right. So that kind of homology based approach works here, but like human chimpanzee can also be done like that, or has been done like that, but it may fail in case of very distant genomes, right. So like prokaryotic genes, if you see, like it is a circular chromosome, and it starts with origin of application and ends at a termination site, and okay, in the chromosome the genes are arranged in the forward strand and the reverse strand, both strands, okay. There could be pseudo genes also, okay. This is the GC content and GC skew which is shown. So many things you can show on the circular map of a prokaryotic genome, but to note here is that there is not much space in between the genes. For example, this is the reverse strand. So basically forward strand is containing gene, reverse is not in that region. Okay, so if you see, like, the DNA is fully occupied with genes, and almost 85 to 90% region is only genes, and remaining could be the intergenic region or regulatory region in between two genes, right? So that is, and they like to keep it very dense, so that it generally helps in their fast replication and transformation of information. Okay, and another important thing about prokaryotes is that their genes may be arranged in a operon, where they form a polycistronic transcript, which is later converted into multiple proteins, right? Introns are not there, only one gene and one protein, because there is no alternate splicing. So it is one gene, one protein concept, that one gene will form only one protein, right? And similarly, like, there is always one open editing frame per gene, and this also holds true for the eukaryotes. Okay, and the start codons are 3: ATG, GTG, and TTG. Generally, like, ATG is more frequent, GTG sometimes, UTTG are rare. Similarly, there are 3 stop codons which can be used to terminate the protein, right? Regarding the gene structure of prokaryotic gene, you can see that if the start of gene is this, like ATG is present somewhere here, right? So upstream of that -10 region, this is basically the promoter, or you can say Pribno box, okay, or basically the Tata region, right, Tata box. So you can see that it has a kind of consensus or conserved TAT sequence, of which T generally is found 79% of the time, A is found 95% of the time, similarly last T is found 96% of the time, and in between are like 51%. So basically it is conserved but yet variable depending upon the species and its GC composition. Okay, similarly there is a -30 region, which is also important for the binding of transcription factors, right? So together, like this Gilbert box, Pribno box, RBS, which is like +10, because polymerase takes position within the RNA, right? So those are important signals for the prediction of prokaryotic genes. So just have a look at the different genome sizes and their ORFs which are known. So you can see that the size of genome is very small, right, and the number of ORFs are also small, and basically keep on increasing with respect to the genome size. And you can see that almost like 1000 base pair will contain 1 MB will contain 1 million base pairs will contain about 1000 genes. So generally it is this kind of ratio. Okay, so for different, like, it could be symbiotic, the number of genes are less. Free living microbes, like, number of genes has to be like more than the symbiotic or endosymbiotic, because they have to do all the functions on their own, or maybe dependent upon environment also for some nutrients, but mostly they are free living, right? So the genes could be as high as 8000, right, or as low as 400. So this also gives an indication of the minimal set of gene that is important for life, or for life to exist. Now for a given genome, for example, this is an example from Archobacter genome. You can see that the ORFs that were predicted are like this. So ORF1 starting from 1 to 1317, located on the positive strand. This is the annotation that was done, the functional category ID, right? Okay, so this way you can arrange from start to end, you can arrange genes in forward and reverse strands, right? And in this case, basically since it is reverse complement, the end came first and start came late in terms of their location on the forward DNA, right? We always report the forward DNA, we now report the double stranded DNA in terms of sequence, because the other strand is kind of obvious, and you can always reverse complement the forward strand to get the reverse strand. Okay, so the common gene finders in prokaryotes include the ORF finder, which uses an ORF based approach and available at NCBI. We will discuss this, right? It's a very simple way of finding genes, but it may find a lot of genes, and it may not follow very sophisticated criteria of finding the exact start or other features of a gene. Glimmer is another one which has been the gold standard from several years to predict the prokaryotic genes, and Prodigal has recently come in the last decade, which is also gaining a lot of importance in predicting the genes, prokaryotic genes. Okay, so basically the approach of ORF finder is very simple, like as we discussed earlier in this lecture also, that it wants to find all the genes or all the regions which have a start codon and in between codons, or basically open reading frame, which is sufficient enough to be called a gene, right? So it identifies every stop codon in the genomic sequence. For example, this is your sequence. So it will, what it will do, it will first identify all the stop codons in the sequence, right? Okay, and upstream of these stop codons, it will find the start codon. Okay, the farthest start codon. Basically, there could be one here, there could be one ATG here, or TTG here, or here, right? Possible. Okay, but it will find the farthest one, and later on it can be resolved which is the correct start and end based on similarity or other factors like promoters, locating promoters, or RBS, right? So generally it looks for ATG, but GTG and TTG are also like 95% you can see find ATG as the start codon, but GTG 4% or TTG 1% in the prokaryotic genome also, right? And there is a concept of minimal length, that it could be that there are two stop codons very near to each other, so this region cannot be an ORF because it is not long enough to code for a relevant protein. Okay, so the ORF finder can be run using NCBI, and you can give your query or upload your sequence here. For example, for this sequence I can select this, and for the which it can run a sample run for the to find the ORFs, and then you can select other parameters such as you may want to find gene in a particular region in that sequence because it could be long genome, or you can specify that what should the minimum length of ORF, like it's here it is 75 base pair, that means about 25 amino acids, 75 divided by 3. Okay, genetic code standard, it could be a species or basically a class specific or mitochondrial or human or like that, or standard is generally used. What should be the ORF start codons? It could be ATG and other alternate also like TTG or GTG. Okay, and ignore the nested ORFs, because if there are too many ORFs in the same region, you may like to ignore and get only the clear or clean ORF. Then you say submit, and it will give you for that query this result where it shows that in the total length of DNA, at what locations you have got the ORF. This sign means in the forward direction, this sign means in the reverse direction. This is your description that you can click on any of these, okay, and get it sequence in this box, right? And basically in this region you can, the genes that you found are mentioned here, ORF number, strand, forward or reverse, frame that matters, that in the DNA in what frame you got the gene, 1 2 3 and then 4 5 6, okay, or +1 +2 +3 and -1 -2 -3 like that, right? The start and the stop and total length of nucleotides and the amino acid it is coding for. So you can see that these are all long genes and should definitely be a gene because or should be basically identified as ORF because these are long enough to code for a protein which is like above 50 amino acid. You can say whatever you find should be a gene because that is long enough to code for a functional protein. Okay, so basically advantage of ORF is that it can find each and every ORF that may exist in a genome or in a given sequence. So basically it is definitely going to over predict, and not all the ORF that it has found will be genes, okay, but it may be helpful because it can find those ORFs which are may be missed by other software like Glimmer or Prodigal, right? Because they might be looking for something and giving a score, and score wise like that got missed or skipped, right? It could not get that high score, but it could be an ORF. And also in case of orthologous ORFs, the genes which have just transferred, those may qualify the ORF finding criteria, but may fail in Glimmer and other software, right? So that is the basically issues with ORF finder and advantages also. Now for like ORF finder you can use, but generally people use like Glimmer or Prodigal or these, or GeneMark, these kind of software, because in addition to ORF finding, they also try to confirm, or the like the ORF that which they are calling a gene is basically very reliable. Its start and end are well defined. They have mapped the other things like promoters, RBS in that ORF, and also looked at the GC composition and codon composition of that genome before calling an ORF. So that is why like they prefer to use Markov models or these kind of machine learning models to first predict and then to verify that gene, right? So Glimmer is a very old software and still like very widely used. Even NCBI uses Glimmer for the validation of genome submitted at NCBI, right? So it can be downloaded from this site, and it was developed by Salzburg, right? Steven Salzburg. And so I will discuss like how it works and what is the algorithm, but this is the main page of Glimmer. And for installation it is simple. You download this file, Glimmer version 3.02 tar.gz, okay, and then tar minus xvf Glimmer. So basically zxvf will open this file and create a directory called Glimmer this, okay. In that directory go to the src folder, cd src, okay, and in that folder just go, because that contains the source or binaries, just say make, then it will compile this for your system, and then the program is ready to use. And basically the program will be in Glimmer 3.2/bin, that is the binaries. Now to run the program, for example there is a file called genome.seq, okay. So there is a script, that is a wrapper script that run 4 different scripts, and you don't have to run 4 one by one. So you can say like G3, okay, Glimmer 3 from scratch.csh. .css is for a script, okay. From scratch means that it is doing an ab initio prediction from scratch. It doesn't want any kind of reference or supporting genes file to see that okay, these are the genes which should be give you like some help in training or something. So it is ab initio, it gets trained on its own on the given genome and does not need any training set from outside. And this is the input file, okay, and the folder, output folder name run1, okay. And then it runs 4 commands, long ORF, extract, build and Glimmer 3, okay, the results of which will be shown in the next page, okay. So basically Glimmer 3 is a specific or highly useful gene finder in prokaryotes, okay, and also in other smaller genomes including archaea and viruses, okay. It is based on interpolated Markov model, and basically and also uses the RBS and promoter information to refine the genes, okay. It also uses the codon bias or GC content of the genome, and uses a nth order Markov model to the genes that it is for making the predictions, right. So like for example, the first thing that it does is from any genome, right, it basically it is always true that if I sort all the genes in like in a genome in the order of decreasing order of length, so the longest genes in any genome will most likely be the genes, right, because bacterial genome do not waste space and they cannot keep a long region which is coding and in its genome and does not use it, or does not is not a basically a coding or functional protein coding region in that genome, right. So Glimmer also uses this approach. So what it does is, using simple ORF based approach, it finds the 500 longest genes from a genome. Okay, so now those 500 genes act as a training set, okay, because that will tell the codon composition of the genome, the type of gene length that is followed in that genome, right. And using those set of long ORFs, it trains its IMMs, and then using basically the nth order of Markov model, basically it is mostly the 8th order, right. That means 9 nucleotides starts from 0 to 8 is basically 0, 1, 2, 3, 4, and like that 8 is 9 codons it is using for making the prediction, right. And then for every ORF that it finds using that training, okay, on the real genome, so it also ranks that ORF by giving a score, okay, which is the likelihood that what could be the probability that it could be a gene, right. And generally, like, it is highly accurate, more than 98 or 98%, and also give you the correct start end, and because that is also an important task when you are doing the gene prediction, right. So the output for the program that we run was, this is the ORF number, start end, strand, and the score that Glimmer did. So the 4 columns are like mentioned here, the description, but I have already told the ORF number, start end, and the reading frame and the score. Another tool that is recently very commonly used is Prodigal, which is very fast, and basically in 10 minutes it can do several predictions, including the gene prediction for a genome, right. And it uses an unsupervised machine learning algorithm, okay. So basically it also uses like the ribosomal binding site, codon statistics for its training. It is available here, and this is the paper that mentioned the details about the Prodigal program. Okay, this is the GitHub site from which you can download the program, and basically the installation download is typical of all the software, and follow the similar way that we have discussed so many times in these lectures. Okay, so basically for running this, you can say Prodigal -i input of your genome, output file genes and proteins. So you want the gene sequences in nucleotide format and protein sequences amino acid format. So and the list of genes. So that is the output, and output also be used for metagenome the same way. Okay, and for installing, you can same like download the program, then say make install, right. And also you can specify the directory in which you want to install, okay. And then just do make, either specify directory and do make install, or just do make. And after installing, basically it is it is doing a lot of jobs for your submitted sequence, that is predicting the protein coding genes, okay, including the GFF format, GenBank format, or also the protein files, or the amino acid nucleotide files for the genes. It can do for draft genomes or metagenomes, even partial genomes can be given. Okay, it is very fast, it is runs unsupervised and does not need a training set. Okay, it can predict the partial genes also, and also predicts the correct start end, and or the correct start codon, because there could be many start codons upstream, a stop codon, and to find which one will be preferred is or this the correct start codon is not so easy. Okay, so if you say Prodigal -help, it will give you all these options, and you can use a combination of these options to refine your gene lengths. Okay, so as I mentioned earlier, basically it is looking at all the information, including the long genes, ORFs, GC content, length of bases, type of codons. Okay, using the hexamers that is derived from that sequence, right? And then scoring, giving score for each and every gene. Okay, so for a genome, say Mycobacterium tuberculosis fasta, so it is that is your input, okay, for Prodigal. So it will you can specify one file which is containing the amino acids FAA, okay, one file which is giving the DNA, that is the nucleotide gene sequence in nucleotide format FNA, and one output directory which is Mycobacterium tuberculosis say out, which contains like your output files including the gene and protein sequence and other files that it found from the genome, okay. And it is very fast, so very helpful for metagenomic and if you are dealing with a lot of genomes, right? So this, for example, was the input file for Mycobacterium, and it gave the amino acid files. So basically there is a list of amino acids like this, and the genes that it predicted from the genome, okay, in the nucleotide formats, okay. And basically the other feature that I have mentioned in between also in this lecture, the promoter and ribosomal binding site could also be of a lot of importance while fine tuning or refining the predicted genes and or giving weightage to the gene, and that have been used by these software, okay. So typically for a new genome or a prokaryotic genome, first of all, like if you have sequenced it, then you have to assemble it, that we have discussed in the previous class, and then you have to take that RAT assembly or basically or the complete assembly if you have. You may like to fill the gaps or as much as possible if you can, otherwise a good draft can also be an input. Then predict the genes using Glimmer or Prodigal or any other software, okay. Then basically you also should look at the origin of replication, that what is the origin of replication gene in that list of genes, and you may like to rearrange all the genes by keeping the origin of replication or ORI genes or chromosome DNA A gene, right, as the first gene in your genome, and rearrange all the genes, because basically it is like a kind of circle with say many wheels, okay. So like this is a wheel, right? So you don't know which one is the first spoke. So it could be like your origin is here, so you may like to give this as number 1 and then arrange all the genes as number 2, number 3, number 4, number 5, number 6, like that, okay. So that becomes the start of your genome. So you may like to find the origin of replication gene first and then rearrange all the genes. Then the genes that you have found, you would like to confirm that they are real genes and not pseudogenes or artifacts, so by BLAST or other software that we will see today, okay. And then also may like to correct the start end of the genes. Generally programs also do this, otherwise RBS Finder, these programs can also be used for this purpose. You should remove or correctly identify the pseudogenes from the analysis, later analysis that you are going to do, right. And then prepare a circular map of the genome like I have mentioned here, right. So basically in the first part of this lecture we have covered the commonly used gene prediction approaches and the methods including ORF Finder, Glimmer, Prodigal, right, and the criteria of finding these genes. And in the second part we will be dealing with the how to annotate the genes that you have found, and also predict the transfer RNA, ribosomal RNA and operons in your sequences. So this is good for this part. Thank you.